Transcription and translation is the process of creating proteins from our DNA. First, transcription. This happens in the nucleus if it's a eukaryotic cell. RNA polymerase separates the two strands of DNA and starts adding complementary nucleotides to the RNA sugar phosphate backbone. RNA does not have the nucleotide thymine, which pairs with adenine, so it is replaced with uracil. The DNA is read from the 3' to the 5' end, but the RNA is created in the opposite direction, from the 5' to the 3'. The RNA polymerase knows where to stop reading the DNA due to a stop codon, which tells it to stop reading. A common one is TTGACA. A stop codon is a series of 3 nucleotides which codes for a specific amino acid. This RNA can become ribosomal RNA, which makes up the ribosome, transfer RNA, or tRNA, which carries the correct amino acid to the ribosome as directed by the mRNA, ribozymes, which are RNA enzymes, or, most importantly, which is what we're going to be talking about, messenger RNA, or mRNA. If it is in a eukaryotic cell, it's a eukaryotic cell, it has a nucleus. The MG cap gets added onto the 5' end, and the poly A tail onto the 3' end in the nucleus before being sent to the ribosome to begin translation. Let's talk about translation. The mRNA actually has the same start codon each time, AUG, which codes for methionine, or MET. The ribosome has 3 parts: the exit, or E site, the P, or peptidyl site, and the amino acid, or A site. The mRNA goes into the ribosome, being read and constantly fed in. The tRNA recognizes which amino acid the specific codon codes for and binds to the A site with it, like this. Another tRNA comes to the site, pushing the first tRNA into the P site. Then a third tRNA comes to the A site, which pushes the second tRNA into the P site and the first tRNA into the exit site, where it's ejected out of the ribosome. The first tRNA actually leaves its amino acid, where it binds with the second amino acid, and this process repeats until you have a full protein, which is actually just a set of amino acids. During translation, it also stops when it reaches a stop codon, which is the same as before, TTGACA, but there are other stop codons. There are also 4 different types of mutations. The first is substitution. A substitution within a single letter in a codon gets changed to a different one. The second is insertion. Insertion is when extra base pairs are added in. The third is deletion, and deletion is when sections of base pairs are not added, or deleted. And the last one is frame shifts. That is when only one base pair gets deleted, which shifts all the codons over 1 when it is to be parsed. This has been Masha talking about how you make your proteins.